Dear students, today we are going to study some important works which have been done in the field of microbiology and associated areas. First of all, a very important antibiotic called as penicillin was discovered by Fleming, Chain, and Florey. There is an interesting story behind the discovery of antibiotic penicillin. It was first of all discovered by Sir Alexander Fleming. He was a Scottish researcher and he was not experimenting on fungi or Penicillium notatum. Rather, he was researching influenza virus. The penicillin antibiotic is the first naturally occurring antibiotic which was discovered in those days, and it was an accidental discovery that Fleming was regarded as a careless worker. He was a leg technician and he went on a holiday of 2-3 weeks. And after spending the vacation, he came back and found that a mold had developed on the petri dish, accidentally contaminating the Staphylococcus culture plate. He then examined that culture plate. Upon examination, he found that he noticed that the culture was prevented. The culture of Staphylococcus was prevented by the growth of the mold, and actually the mold was lysing the bacterial colony. Means he derived a conclusion that some bactericidal and bacteriolytic properties uh, is bestowed in the fungi which is preventing its growth. That led to the discovery of this very important antibiotic, and this has helped reduce the number of deaths and amputations of limbs in the troops during the World War II. Unfortunately, Fleming stopped researching on penicillin after 1931, and the story which I had narrated before you was happened in the year 1928. But somehow he stopped in the work in 1931, but his work was carried forward by Howard Florey and Ernest Chain. This penicillin antibiotic is effective against gram-negative bacteria, ineffective against gram-negative bacteria, but very effective against the gram-positive bacteria. So this is the story behind the discovery of a very important antibiotic called as penicillin. Then came M. Thiele in 1951. He discovered vaccine for yellow fever and got Nobel Prize. Enders, Wellers and Robbins in 1954 able to culture poliovirus in tissue. You can only culture the viruses in the tissue, as you know that they function only in the living system, or they require host cell to replicate. Then in 1957, Bowe came and he discovered antihistamine. Histamine, as you know, is an inflammatory substance mainly secreted by mast cells and some other cells of the body also. He also got Nobel Prize for his discovery. Then came Beadle, Totham and Lederberg in 1958. They worked very hard on genetics, and they were associated with microbial genetics, and they got Nobel Prize too. Okoa Korenberg in 1959 discovered enzyme catalyzing nucleic acid synthesis. That means the DNA and RNA polymerases. For that, they got Nobel. Watson and Crick in 1962, they made many discoveries related to the structure of DNA. You know that the DNA contains double strand and that is anti-parallel, and all the dimensions have been worked out by Watson and Crick. Then came Jacob and Monod in 1965. They worked on gene regulation, that the gene do not remain switched on forever but can be regulated. They can be made to run or made to produce their products, or can be switched on as per requirements or as per desire. So Jacob and Monod gave operon model, and they found a very important uh, discovery related to gene regulation. Rouse in 1966 discovered cancer causing viruses, the oncoviruses. Then came Holly, Hargobind Khorana and Nierenberg in 1968, and their work was very important. They deciphered the genetic code. That means they discovered that the genetic code is triplet, and each is called as codon, codes for a single amino acid, as you might have read when you have gone through the translation. Temin and Baltimore discovered RNA dependent DNA polymerase, which is also called as reverse transcriptase, and in RNA tumor causing viruses. This RNA invariably undergoes an intermediate stage which has DNA. Then came Blumber and Gajusek in 1978. They developed the mechanism and dissemination of hepatitis B virus. Benaklevaf, Snell, and Dowsett in 1980 discovered histocompatibility antigens. Gilbert Sanger in 1981 discovered DNA methodology of DNA sequencing. Then came Milstein, Kohler, and Jern in 1984. They developed technique for formation of monoclonal antibodies, and these monoclonal antibodies derived from they are actually produced by identical immune cells, which are actually clones, and all bind to same epitope. Ruska invented transmission electron microscope in 1986. This was also a very important invention. Then came Bishop Warmus, who discovered the oncogenes, and they said that the oncogenes are created in the genome by proto-oncogenes due to certain mutagens. Altman in 1989 discovered ribozymes, which are non-proteins and non-protein enzymes. Then came Mullis and Smith in 1993. They developed polymerase chain reaction, which is used these days to detect the COVID infections. Then came Prusiner in 1997. He discovered prions, as I have discussed in the previous lecture, that prions are purely protein particles and they do not contain any nucleic acid, but instead of that, despite that, they could able to cause disease in human beings and other animals also. Then came Nathan Arbor in 1978. They discovered another an important enzyme called as restriction enzyme, which is a molecular scissor. That means it cuts the nucleic acid at a size precisely located or somewhat away, a few nucleotides away from the desired site. These restriction enzymes, they play a very important role in today's modern genetic engineering and biotechnology for cutting the genes. uh, these are some of the important discoveries and inventions, and all of them, they got Nobel prizes. And now we come to the microbiology institutes in India which are working in the field of microbiology. First one is TERI, which is Tata Energy Research Institute, situated at Delhi, working in the field of microbiology. Uh, they developed oil zappers, which is a consortium of bacteria which degrades the polluted sites which are polluted by the spill of oils in oceans. Then National Institute of Nutrition, also working in this field, is situated in Hyderabad. Then National Chemical Laboratory is situated in Pune. National Institute of Science situated in Bangalore, and NEERI is situated in Nagpur, which is National Environmental Engineering Research Institute. Then lastly, we have National Institute of Occupational Health situated at Ahmedabad. So students, this is all in this lecture. Thank you very much.